Welcome back to the online class, students. In the previous class, we were discussing about the nucleic acids and the types. Okay, so in which we have studied about the DNA. So what are the DNAs are made up of? Components of DNA and the double helix structure of DNA we studied in the previous class. In today's class, we are going to discuss with the another nucleic acid, that is the ribose nucleic acid. Okay, so nothing but RNA. Okay, so the RNA is nothing but, as we know, it is a type of nucleic acid and it is found in case of cytoplasm predominantly, and even it can be found in the nucleus. Okay, so the ribonucleic acid is a type of nucleic acid. It is found in case of where? It is found in case of cytoplasm and nucleus. Cytoplasm and nucleus. Preferably, major amount it is found in case of the cytoplasm. And this RNA, so the nucleic acids like the DNA can found as the genetic material in most of the living organisms, and RNA can be found as a genetic material only in case of few viruses. Okay, so the RNA also act as what? Genetic material in case of only few virus like so plant and animal virus, for example TMV. So TMV is nothing but tobacco mosaic virus. Okay. So other than that, in case of eukaryotes, the RNA is found in case of cytoplasm and in case of a nucleus. Where? So this RNA is produced by the DNA by a process called as transcription. Okay. So RNA is produced from what? DNA. RNA is produced from DNA. The process is called as transcription. The process is called as what? Transcription. And so the RNAs are, structurally when you consider this RNAs, they are single-stranded. Okay. So the RNA is made up of only one polynucleotide chain. They are single-stranded structures. It means they are made up of only one polynucleotide chain. Okay. So they are made up of only one polynucleotide chain. Okay. Then, so as we know that, so in case of most of the virus, they act as a genetic material. But whereas in case of eukaryotes, this RNA can function or help in protein synthesis. Okay. So in case of eukaryotes, so the RNA helps in what? Protein synthesis. Okay. So this is regarding the introduction of RNA. So as I told you that RNA is single-stranded. It is made up of only one polynucleotide chain. So poly means what? Many. And so it is made up of what? Nucleotides. Okay. So they are made up of nucleotides. That means many nucleotides are linked with one another to form a large structure called as what? Polynucleotides. Okay. So let us know what are the nucleotides. Already we have discussed about this. Okay. So the nucleotide means it is made up of how many components? It is made up of 3 components. Which is that? Pentose sugar, nitrogenous base and phosphate. Okay. When you consider a nucleotide, it is made up of 3 common components. Preferably, it is pentose sugar. It is a pentose sugar, phosphate, and nitrogenous base. Nitrogenous base. So let us study one by one. So the pentose sugar, as the name suggests, it is made up of 5 carbon atoms. The pentose sugar found in case of RNA is ribose sugar. Okay. So the pentose sugar found in case of RNA is the ribose sugar. Hence the name RNA is nothing but ribose nucleic acid. Okay. So ribose sugar, let us see the structure of ribose sugar here. So the ribose sugar made up of how many carbon atoms? 5 carbon atoms. when you see the general structure, it is C5H10O5. C5H10O5. So it is a pentose sugar, isn't it? So here it will have a pentagon-like structure like this. Okay. So where 4 carbon atoms are found inside the ring, 1 carbon atom is found outside the ring. Okay. So that is carbon number 1, 2, 3, 4 and 5th carbon atom will be found here. Okay. And coming to the functional groups. So it will have OH, OH, OH here and CH2OH on the 5th carbon atom. Okay. So this is the structure of the ribose sugar which is found in case of the RNA. So and here you can notice that so the second carbon atom is having the functional group is OH. Okay. Second carbon atom is having the functional group is what? OH. So here when compared to deoxyribose sugar, what is deoxyribose sugar? So it is nothing but C5H10O4 where so here second carbon atom will not have any OH. Instead of that it will have only hydrogen, isn't it? So here you can find that it is a pentagon-like structure. This is also so 4 carbon atoms are found inside the ring and the 5th carbon atom is found outside the ring. So here is the OH. So second carbon atom will have only H. Third carbon atom will have OH and 5th carbon atom will have CH2OH. So first, second, third, fourth and 5th. See here. So due to the more number of functional groups or ribose sugar, so this is having more reactivity. Okay, or it is less stable. So and due to the less number of functional groups, that is you can see that on the second carbon atom you will not find any OH, only H is present. Due to that, so it gives more stability to the DNA. Hence, DNA is found as a genetic material in most of the living organism. This is also one of the reasons. Okay, so can you make out the difference between the ribose and the deoxyribose? So deoxyribose, as it is deficient of one oxygen, we are naming this as the deoxyribose sugar. And here in the second carbon atom, you can find the OH. So this is the ribose sugar. Is it? Next one is phosphate. So phosphate is nothing but they are the derivatives of phosphoric acid. Phosphate is what? They are the derivatives of phosphoric acid. So where phosphate formula is PO4, where 1 phosphate and 4 oxygen atoms combines together forms the phosphate. So phosphate is made up of what? They are the derivatives. They are the derivatives of phosphoric acid. They are the derivatives of phosphoric acid where phosphate is made up of 1 phosphate atom and 4 oxygen atoms together form the phosphate. Okay. So let us study about the nitrogenous base now. Okay. So already we have studied about nitrogenous base in case of DNA. So what is a nitrogenous base? They are the nitrogen containing compounds. Okay. Nitrogenous bases are nitrogen containing compounds. So and these nitrogenous bases will have a heterocyclic ring-like structure. Okay. So nitrogenous bases, nitrogenous base, they have a heterocyclic ring-like structure. Okay. So I can define nitrogenous bases as, so they are the complex nitrogen containing heterocyclic compounds. Okay. So nitrogen bases are defined as, they are the complex nitrogen containing heterocyclic compounds. Okay. So here the nitrogenous bases in case of RNA, it is important, remember that. So in case of RNA, so there are majorly 2 types, that is purines and pyrimidines. Okay. The 2 types of nitrogenous bases are purines and pyrimidines. Purines and pyrimidines. What are purines? Why we have to divide them into purines and pyrimidines? Based on the number of rings, we can divide them into purines and pyrimids. Purines have two ring-like structure, and they are the double ring-like structures. Pyrimidines are made up of only one ring, that is single ring-like structure. So they are made up of only one ring-like structures. Okay. So which are the purines? The purines are adenine and guanine. Okay. So the purines are adenine, represented by capital letter A, guanine, represented by capital letter G. Okay. Then coming to the pyrimidine. So pyrimidines are of two types, that is cytosine and uracil. Okay. So that is cytosine and uracil. So cytosine is denoted by capital letter C. Uracil is denoted by capital letter U. And the most important one is uracil is a nitrogenous base found only in case of RNA. Okay. So uracil is found only in case of RNA. In case of DNA, in the place of uracil, you will find thymine. Isn't it? So these are the different types of nitrogenous bases found in case of the RNA. Okay. So now already we have discussed the structure of adenine, guanine and cytosine. So let us see the structure of uracil. So as I told that uracil is an example for pyramid. So it is a single ring-like structure. Isn't it? So now let me write the structure of uracil. NH, double bond, NH. Okay, then functional groups here are O, double bond O, double bond O. So this is the structure of uracil. Okay, so this is regarding nitrogenous bases. As we know that, so this is also one of the type of nucleic acid. Okay, so the nucleic acids are linked with, in nucleic acids, nucleotides are linked with one another with the help of what? The bond called as phosphodiester bond. So as we saw the phosphodiester bond formation in the beginning itself, so the phosphate links the third carbon atom of, so pentose sugar of one nucleotide with the fifth carbon atom of pentose sugar of another nucleotide. We call it as what? Phosphodiester bond. Okay. So now let us see which are the different types of nucleosides which are present here. Okay. So the different types of nucleosides which are present here. Let us see that. Then we will study the different types of nucleotides present in the RNA. Okay. So first one is nucleosides. The nucleosides present in the RNA. Okay. So before discussing nucleoside, what is a nucleotide? Okay, so now we are going to discuss about nucleoside. Okay, so we know that nucleotide means it is made up of three components, that is pentose sugar, phosphate and nitrogenous base. But here nucleoside is formed by two components, that is pentose sugar combines with nitrogenous base. Okay, here what happens here? Pentose sugar combines with nitrogenous base. Okay. So pentose sugar, let me write the nitrogenous base first. So combines with nitrogenous base. Pentose sugar combining with nitrogenous base results in the formation of nucleoside. Okay. So as we know that, so there are the four different types of nitrogenous bases are found in case of RNA. Which are those? That is adenine, guanine, cytosine, uracil. Isn't it? Now, which is the pentose sugar found in case of RNA? That is ribose sugar. Okay. So, the pentose sugar found here is the ribose sugar. Okay. Now, adenine combines with the ribose sugar and forms into a nucleoside called as adenosine. Okay. Adenosine. So adenosine is formed by when Adenine combines with the ribose sugar, it forms into adenosine. Sorry. Yes, adenosine. Okay. Then next, guanine combines with the ribose sugar, results in the formation of guanosine. Okay. Guanine combines with the ribose sugar to form guanosine. Then next, cytosine combines with the ribose sugar and forms into a nucleoside called as cytidine. Nucleoside called as what? Cytidine. Okay. The next one is uracil. So uracil combines with the ribose sugar to form uridine. Okay. To form what? Uridine. Okay. So this is regarding the 4 nucleosides which you can find in case of the RNA. Now let us see about nucleotides. How the nucleotides are formed. So now let us discuss about the nucleotide. So as you know that nucleotide means it is made up of how many components? 3 components. Is it? So that is pentose sugar, nitrogenous base and phosphate. Which is that? Pentose sugar, nitrogenous base and phosphate. Isn't it? So now, nitrogenous base and pentose sugar together, what we call nitrogenous base and pentose sugar together forms nucleoside. So can I call this as nucleoside? Okay. Nucleoside combines with phosphate and results in the formation of what? Nucleotide. Okay. So let us see that. So how the nucleotides are formed. So first let me write the different types of nucleosides here. That is adenosine, guanosine, cytidine, uridine. Okay. So these are the 4 nucleosides found in case of RNA. Okay, now, so this nucleoside combines with what? Phosphate and forms into a nucleotide. So let us write the nucleotide. Adenosine combines with phosphate and results in the formation of adenosine. How many phosphate? 1 phosphate. So monophosphate. Okay, so the nucleotide formed here is adenosine monophosphate. Okay. So in short form, we'll call this as AMP. Okay. So on this AMP, in short form it is called as AMP. So this AMP act as a secondary messengers. Okay. So in case of hormone controlled reactions. Okay. The next is guanosine combines with phosphate. So to form guanosine monophosphate. Okay. Guanosine monophosphate. Okay. The next is cytidine combines with phosphate to form cytidine. How many phosphate? 1. Monophosphate. Okay. So in short form, we write this as a CMP. So guanosine monophosphate as GMP. Okay. The next is uridine combines with phosphate to form uridine monophosphate. Uridine monophosphate. So this can be written as in short form UMP. Okay. So these are the 4 different types of nucleotides found in case of the RNA. Okay. So some of the higher nucleotide means, so when nucleotides, so there is addition of some more phosphate group, it acts as a energy carriers. Okay. So as you know that, for example, ATP. So it stands for adenosine triphosphate. So when 3 phosphate groups are found, so there is a high amount of energy can be carried with the help of the nucleotides. Okay. So higher nucleotides means which has more number of phosphate, they can act as a energy carriers. As you know that One of the important one is ATP, adenosine triphosphate. Adenosine triphosphate. Okay, so this is also called as energy currencies of the cell. So this is regarding the nucleotides found in case of the RNA. So now let us study the types of RNA, which is most important. Okay, types of RNA. So actually there are many different types of RNAs are there. So majorly, so the RNAs are classified into three types. Majorly the RNAs are classified into three types based on their physico-chemical nature and functions. Next one is classification. Under classification, they are classified based on what? Physico-chemical nature and their functions. Okay, and their functions we have majorly three types, that is mRNA, tRNA and rRNA. Okay, so which are the three types here? That is mRNA, okay, tRNA and rRNA. Okay, so these are the three different types. Let us see one by one. Okay, so first let us discuss about the mRNA. Later we'll discuss about tRNA and rRNA. So mRNA stands for messenger RNA. mRNA means messenger RNA. Okay, so this mRNA accounts for 8% of the total RNA. Okay, it accounts for 8% or 5 to 10% of the total RNA. So now, this messenger RNA, so as we know that RNAs are synthesized by the DNA by a process called as transcription. So this mRNA is synthesized by DNA. So with the help of an enzyme called as RNA polymerase. Okay, so with the help of enzyme called as what? RNA polymerase. So here the RNA polymerase is 2. RNA polymerase second helps in the synthesis of the mRNA. Okay, now, so the function of mRNA is, so this carries the information from the DNA for protein synthesis. Hence, it is also called by another name as informational RNA. Okay, it is called as informational RNA as it carries the information. As it carries the information from where to where? From DNA to the ribosomes. From the DNA to the ribosomes for protein synthesis. For protein synthesis, hence it is also called as what? Informational RNA. Is it understood? Okay. Next is, so the information is carried, we know. So how that information is carried? So the information is carried in the sequence of 3 nucleotides. Already we know that what is a nucleotide, isn't it? So the information is carried in the sequence of 3 nucleotides. The sequence of 3 nucleotides. This 3 nucleotides is called as triplet codon. Okay, it is called as what? Triplet codon. Okay, is it clear now? Okay. Then mRNA in case of eukaryotes. Eukaryotes means you know that which will have a definite nucleus. So in case of eukaryotes, mRNA is called as monocistronic. It is called as what? Monocistronic. What is the meaning of monocistronic? That means mRNA carries the information only for synthesis of only one protein. Okay, synthesis of only one protein. Such type of mRNA is called as what? Monocistronic. Okay, next in case of prokaryotes. So prokaryotes means which is not having the definite nucleus. So in such cases where the mRNA is called as polycistronic. mRNA is called as what? Polycistronic. So as you know that already you know the meaning of poly. Okay. So here the mRNA carries information for synthesis of more than one protein or many proteins. Such type of mRNA is called as polycistronic, which is found in case of prokaryotes. So this is regarding the mRNA. So let us study about the tRNA. Okay. So tRNA. Now the tRNA full form is, it is transfer RNA. Okay. tRNA stands for transferred RNA. Okay. So tRNA accounts for 12% of the total RNA. Okay, it accounts for 12% of the total RNA. And the tRNA is synthesized by the DNA with the help of an enzyme called as RNA polymerase. RNA polymerase 3. Okay. The next, so this tRNA is also called as, so this is the soluble RNA. So as it is soluble in, so 1 molar NaCl, so the tRNA is also commonly called as soluble RNA. And among all the three, so this is the smaller in size. Okay, so this is the smaller RNA. So this is the smaller RNA. And it is also called as adapter RNA or adapter molecule. So this is called as adapter RNA because DNA has the capacity to carry the amino acids from the cytoplasm towards the ribosome. Hence, they are called as transfer RNA, or it is also called as adapter RNA. Okay, what it does? It transfers the amino acids from where? From the cytoplasm towards ribosomes, which are called as protein factories. Okay. Now, so as we know that, so in case of protein synthesis, there are 20 different types of amino acids are involved. Okay. So to carrying this 20 different types of amino acids, so there are different types of tRNAs are found in the cytoplasm. Is it clear, students? Okay. Then, when you consider the structure of tRNA, it is having a clover leaf-like structure, or the three-dimensional structure is a L-shaped structure. Okay, is it clear? So, tRNA has a clover leaf-like structure. That is, it is actually RNA is a single-stranded. So, here it is folded to form a clover leaf-like structure. Okay, so like this. The clover leaf-like structure will be formed. You can find the 4 arms here. Okay. So these are the 4 arms of the tRNA. Okay. So here the 4 arms are, they have been named as, so first one, this is the anticodon end. So this is the anticodon end where the mRNA will bind. So where the mRNA binds. And opposite to that is the amino acid binding end, amino acid binding site. Okay, so that amino acid binds. Okay, the next is on either side you can find the loops. So one is called as DHU loop. DHU means dihydrouracil loop. Okay, dihydrouracil loop. On another end you will find pseudo-uridine group. Sorry, loop. Pseudo-uridine loop. Okay, so this can be written as in the simple form like this. Okay, pseudo-uridine loop. And in some tRNAs, you will also find one extra arm. So like this, one extra arm also will be found. So extra arm. Okay, so as you know that anticodon binds with the mRNA. So amino acid binding site binds to the amino acid, whereas DHA loop can bind to the amino acid activating enzyme. And so pseudo-uridine group can recognize the ribosomes or helps in the binding with the ribosomes. Okay. So and in case of RNA, just it will have a linear sequence like this. Linear sequence of nucleotide A, U, G, C, A like this. Okay. This is a linear structure mRNA. Okay. The next is rRNA. So rRNA stands for ribosomal RNA. So it is nothing but what? Ribosomal RNA. So as it is abundantly found in the ribosomes. Okay. So this is the large RNA. So this is the large RNA. So the function of RNA is it helps in attachment of mRNA to the ribosomes. What is the function of rRNA? It helps the mRNA carrying information for proteins to attach to the ribosomes. Hence, it helps in synthesis of proteins. Okay. So, what is the function of rRNA? It helps in attachment of mRNA to the ribosomes. Okay. Is it clear now? And so, it accounts for total 80% of the total RNA. So, this is the abundantly found RNA. So, this rRNA, so as it forms the integral part of the ribosomes. Okay, it forms the integral part of the ribosomes. You know that in ribosomes, in case of eukaryotes, we have 80S type of ribosome. In case of prokaryotes, we have 70S type of ribosome. So the S represents Svedberg unit. Okay, S represents Svedberg unit. Okay, so now, so what is that 80S means? So usually this ribosome, they are made up of one large unit and one small unit. So one large unit and one small unit. So this small unit here is made up of 40S type. In case of eukaryotes I am talking. So in case of large unit, it is made up of 60S type. Okay. Students, don't get confused. It is not addition. So this Svedberg's unit means when they are subjected for centrifugation. So at particular level, at particular unit, they will get separated. So that is what it specifies number. So the small unit can separate at 40 Svedberg unit, and large unit can separate at 60 Svedberg unit. When they are together, they can separate at 80 Svedberg unit. So then next, coming to the prokaryotes. So what type of ribosome they have? That is 70S type. So the large unit and the small unit. So here the large unit will have 50S type and here it will have 30S type. Okay. So you have understood the meaning of S, I think. Okay. The last part is, so the functions of the RNA. So as you know that the RNA can function as, so when you consider the biological significance of RNA, RNA can function as genetic material in some of the virus. Okay. Then next, RNA in case of eukaryotes helps in what? Protein synthesis. It helps in protein synthesis. Next, so the RNA, as you know that ribosomal RNA, it forms the integral part of the ribosomes. They forms as what? Integral part of the ribosomes. There are some of the RNAs which function as an enzyme. Usually enzymes are made up of proteins, but here some of the RNA which function as a enzyme, we'll call it as ribozyme. Z-Y-M-E, ribozyme. And not only that, when you consider the synthesis of DNA, so a small unit of RNA should be produced or synthesized first. Okay, so that means it acts as a initiator. Without the synthesis of small segment of RNA, DNA synthesis is not possible. Hence, it acts as an initiator in case of DNA synthesis. It plays an important role even in case of DNA synthesis. So this is regarding some of the biological significance of RNA. So let us see some of the questions related to the today's topic. So first question is, RNA does not possess uracil, thymine, adenine and cytosine. So which is that? Thymine is not found in RNA. It is found only in case of DNA. The answer is B. The next, in 3D view of molecule tRNA-S. So the tRNA molecule, as I told you, so it will have a clover leaf-like structure or L-shaped structure. Okay, the options are L-shaped, S-shaped, Y-shaped, E-shaped. Okay, in 3D dimensional view, it will have a L-shaped. The answer is A. Then, anticodon occurs in tRNA, mRNA, rRNA, DNA. So anticodon occurs in case of a tRNA. So the answer is A. The next is, which RNA forms the integral part of the ribosomes? That is mRNA, rRNA, tRNA, none of this. The answer is B, that is rRNA, that is ribosomal RNA. Okay. So this is regarding today's class, students. Thank you.